A delightful morning to everyone here. To mark today's session an auspicious beginning, we'll start with lamp lighting and Ganesh Vandana.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
I, Jasma Jana, Assistant Professor, Roland Institute of Pharmaceutical Sciences, welcome all the delegates to the 9th day of Series 2, Session 1, Online Faculty Development Program on Emerging Trends in Drug Design and Development, sponsored by AICTE New Delhi and organized by Roland Institute of Pharmaceutical Sciences, Barampur, Odisha. Today we have with us revered resource person Dr. Subrat Kumar Bhattamishra Sir, presently designated as Professor and Head, Department of Life Sciences, International Medical University, Kuala Lumpur, Malaysia. He will be giving his talk on the topic Connection between Insulin and Brain in Alzheimer's Disease. So before Dr. Bhattamishra Sir begins his talk, I take this opportunity to introduce Dr. Bhattamishra Sir to all of us. Dr. Bhattamishra Sir has completed his PhD in Pharmacology from Institute of Technology, Banaras Hindu University, Varanasi in the year 2007. He has done his M.Pharm in Pharmacology from Birla Institute of Technology, Ranchi, India in the year 2001. He has completed his B.Pharm from Barampur University, Odisha in the year 1999. He has also done Professional Diploma in Clinical Research from Catalyst Clinical Services, India in the year 2006. Then he worked as Scientist 1 and Manager from the year 2007 to 2009 in Department of Pharmacology at Torrent Research Center, Ahmedabad, Gujarat, India. After that, he has joined as a Professor and Head in Postgraduate Department of Pharmacology, Roland Institute of Pharmaceutical Sciences, Barampur, Odisha from 2009 to 2012. Then he joined as an Associate Professor from 2012 to 2014 in Faculty of Medicine at Asia Metropolitan University, Malaysia. He then joined as Senior Lecturer in School of Pharmacy, International Medical University, Kuala Lumpur, Malaysia, and he is continuing as Head of the Department of Life Sciences there in IMU University, Malaysia. Dr. Bhattamishra Sir has completed around 20 projects as Principal Investigator and co-researchers funded by Ministry of Higher Education, Malaysia. Worth of projects is around 18 to 30 lakhs. He has published more than 66 national and international publications to its credits. He has guided more than 8 PhDs and 10 PGs to candidates. Dr. Bhattamishra Sir has also adjudicated poster presentation session in 6th International Postgraduate Conference on Pharmaceutical Sciences in the year 2018, jointly organized by IMU Malaysia and University of Technology, Malaysia. He was also invited as an expert speaker on diabetes and obesity by Kalinga TV television channel Orissa on February 25, 2018. He is also invited as guest speaker to Roland Institute of Pharmaceutical Sciences, Barampur, Odisha, uh, to give seminar on introduction to research methodology and workshop on animal ethical committee in the year 2018. Along with this, Dr. Bhattamishra sir is recipient of many honors and awards, like he has received Single Original Research Article Scientific with Highest Impact Factor Award in the year 2020 by IMU Kuala Lumpur, Malaysia. He has also received Best Original Research Paper Award in the year 2019 by IMU Malaysia. He has also received first prize in poster presentation in Diabetes Asia 2017 conference. He has received the Best Poster Presentation Award in International Conference on Recent Advances in Pharmaceutical Sciences, BHU, Varanasi, India. He has received Good Clinical Practice certification by National Committee for Clinical Research, Malaysia. Sir has also received Fundamental of Teaching and Learning license from International Medical University, Malaysia. He has a biographic profile. His biographic profile has been included in Marquis Who's Who in the World in the year 2010 and 2014. He has been also awarded with Senior Research Fellowship by UGC to pursue PhD at Department of Pharmaceutics Institute of Technology, BHU, India. Sir is also lifetime member of APTI, IPA, and Indian Pharmacological Society, India. With this, I now request Sir to kindly come on board and start with today's session.
Thank you so much, Dr. Jaina, for introducing me with a very elaborate information. But actually, I don't need so much elaborate information. This is a simple profile. So I hope I can. Can you hear me all? Is it audible to all of you? Okay, I cannot hear the response. If yes, please chart, at least put a chart on
Yes, sir.
that
Share your screen, sir.
response. Can? Okay, so I'll
Yes,
proceed with my presentation. Okay.
sir.
Okay, thank you so much. So the presentation is visible to all of you.
Yes, clear, sir.
Okay. So good afternoon to everyone, because good morning also. Why I told good afternoon, because here it is afternoon, so I told good afternoon anyhow. So I welcome all of you for this presentations, and I must thanks Dr. Biswaranjan, and Professor BBB Ravi Kumar, Dr. Niranjan, Madam Jailakshmi, and the management of Roland Institute of Pharmaceutical Science. So today's talk will be on the connection between insulin and brain in Alzheimer's disease. So before going to the talk, I will be introducing myself, where I'm working and what university it is. And if you see this picture, this is the building of International Medical University. Basically, this International Medical University started as a international medical college in 1992, and this is the first university, you can say, as the medical and healthcare science product, as healthcare science programs. Okay. So after 7 years of International Medical College, this has been converted to International Medical University. And since then, the International Medical University is offering around 12 undergraduate programs, 14 postgraduate programs, and also it offers MSc, Masters in Research, and PhD by Research. So these are the academic programs which is offered by International Medical University. And International Medical University is one of the premier university in Malaysia, where it offers a lot of research activities, and it is also recently been ranked in QS ranking under pharmacy and pharmacology. The rank is around 300 something, and it has different clusters that is involved in various research activity. So if you see here, it is cancer and stem cell research, and other clusters are bioactive molecules and drug delivery. So I am working under this particular cluster where mostly we are bioactive and bioactive molecules we study, like from natural products and synthetic products, and drug delivery also. And other clusters involve environmental and population health, translational research, and health profession education research. So currently in International Medical University have 500 faculties around, and it has around 4 to 5,000 students. And the students are around over different countries, like around 12 to 15 different countries the students are studying in IMU. At the same time, faculties has also been from different countries, around 10 to 12 different countries they are working in IMU. So this is the brief introduction about the place where I'm working before going to the topic. Now the topic is, as I told, the connection between insulin and brain. So the content of this research of this particular topic includes, first we'll have a discussion on why insulin is important for the body, and what is the role of insulin in brain, what is the source of insulin in the brain, and the link between insulin and Alzheimer's disease pathophysiology. So we'll discuss the link between different pathological hallmark of Alzheimer's disease, and then we will summarize the presentation and we will conclude it. So the presentation may take more than 1 hour, so please be patient with me. So let us start with insulin. What is insulin and why it is important? So I need not to explain much on this particular hormone, because this is a very famous hormone, and everyone knows what is insulin. And why insulin is important, because it is utilize our blood glucose, and it plays a very important role for regulating the blood glucose level. Okay, so the insulin name has actually derived from a word called as insulina. Okay, Latin word actually, insula, and that meaning is the island. So as we know that insulin is secreted from the pancreatic beta cells, which is also called as islets of Langerhans. Okay, so the insula is the Latin word and indicating the island. Okay, so it is a 51 amino acid molecule. Okay, macromolecules protein, and it is secreted from the beta cells of pancreas. Now, what is it we do in peripheral organs? If you see here, the figure describes the role of insulin in peripheral organs, and later we will also discuss in the brain. Okay, what is happening? So let us first discuss about the insulin in the peripheral organs. So I always think insulin is the boss for regulating the blood glucose and also for regulating the metabolic process of our body. How it do all this? So if you see here, insulin actually binds to the insulin receptor, the alpha subunit of the insulin receptor. And after it binds to the alpha subunit of insulin receptor, it undergoes dimerization, and then the beta subunit is activated. So the beta subunit actually it contains tyrosine phosphorylase. So tyrosine phosphorylation takes place. Okay, tyrosine kinase phosphorylation takes place, and due to the phosphorylation of tyrosine kinase, actually it activate the insulin receptor substrate 1 phosphorylation, this IRS1 phosphorylation. And after the phosphorylation of IRS1, actually it stimulate or activate another enzyme that is called as phosphoinositol 3 kinase. Okay, PI3K. And the function of PI3K is it converts PIP3, that is phosphoinositol triphosphate, biphosphate, PIP2, phosphoinositol biphosphate, to inositol triphosphate, IP3. Okay, once the IP3 is formed, then actually it recruits these enzymes, PDK1 and protein kinase B. This PDK1 and protein kinase B phosphorylation is very important in insulin signaling. The phosphorylation of the PKB, or it is also called as AKT, other name of PKB is AKT. Phosphorylation of AKT actually it helps in the translocation of the glucose transporter. So this PKB phosphorylation actually triggers the translocation of glucose transporter from cytoplasm into the cell surface. So if you see here, the glucose transporter is synthesized by the endoplasmic reticulum, and then this transporter is translocated to the cell membrane to actually to function to augment the process of glucose uptake. Okay, so you remember the sequence is phosphorylation of tyrosine kinase, and then phosphorylation of IRS1, and that leads to the activation of PI3K kinase, and then that further stimulate the AKT or the PKB. So this both are same, and due to the phosphorylation of AKT, the GLUT4 translocation takes place, or glucose transporter translocation takes place, and the glucose can be uptake into the cell. So the insulin sensitive organs in the periphery is skeletal muscle, liver, and adipose tissue. So in skeletal muscle, generally the GLUT4 is expressed, and expression of GLUT4 or translocation of GLUT4 at the cell surface causes the glucose to be utilized by the skeletal muscles, and the ATP production takes place. So glucose is utilized as an energy in the skeletal muscle, and for the process of utilization, you need the GLUT4 translocation. Okay, in liver, it is having the glucose, the GLUT2, which is present in the hepatocytes, and that causes glucose uptake, and that helps in the synthesis of glycogen, and also it inhibits the gluconeogenesis. And in adipose tissue, glucose uptake, it causes increased lipogenesis and inhibit the lipolysis. So insulin is a very good anabolic hormone, because insulin actually it helps in the lipogenesis instead of lipolysis. So if you see the persons who is having the actually those who are taking a lot of glucose or carbohydrate, the chances of developing body fat is higher in those persons, because the insulin actually convert the carbohydrate to the lipids and store in the adipose tissue. So this is the mechanism or the insulin signaling in the peripheral organs, and how it is useful for the uptake of glucose and its utilization. Now, what is the role of insulin in brain? Actually, brain is a very small organ compared to our body, isn't it? If you see here, our brain is only 2% of our body, but you know, brain utilizes 20% of the oxygen what we take. So although the brain is very small, but it is a highly metabolic organ. Okay, it is a very highly metabolic organ, and it utilizes a lot of energy, whether it is glucose, and the primary source of energy is glucose. Okay, and there is a clear link between insulin and brain function, which has been already established. And how it is, the link is established that insulin regulates brain glucose metabolism, and insulin also regulates feeding behavior, maintains energy balance, promotes neuronal survival, and also facilitates synaptic plasticity. So insulin has been also reported as the neuroprotective effect in the brain. Now, in the brain, what is the role of insulin? If you see here, this figure shows you the insulin signaling pathway in the brain. Although everything is same, if you see here, it is same. The insulin receptors, phosphorylation of insulin receptors causes iris phosphorylation, and then activate the PI3K, phosphatidyl phosphoinositide 3 kinase. Then the this PI3K convert PIP2 to PIP3, and then PIP3 recruit the PDK1 and stimulate the AKT phosphorylation. So in brain, the stimulation of AKT phosphorylation that causes activation of FOXO. This is called as forkhead family of transcription factor, and it is very important in controlling the energy homeostasis. And FOXO is also involved in metabolism of brain and neuronal proliferation, and this is a very important signaling pathway takes place in the leptin function or leptin sensitivity. And AKT phosphorylation also activate the mTOR, okay, that is a mammalian target of rapamycin. And mTOR is very much useful in synaptic plasticity and also normal neuronal homeostasis. So mTOR is a very important signaling pathway in the autophagy, where actually the autophagy means it actually clear the misfolded protein or degraded protein from the neuron, so that the new protein can be synthesized, or the neurons can be developed. So mTOR is very important role in synaptic plasticity and also autophagy. And AKT also phosphorylate the GSK3 beta. Actually, GSK3 beta phosphorylation causes inactivation of GSK3 beta, and this inactivation of GSK3 beta is very important in the pathophysiology of Alzheimer's disease, because GSK3 beta activation can cause tau phosphorylations, or even it is actually a hyperphosphorylate the tau. So insulin actually it causes the phosphorylation of GSK3 beta, so it inactivate, actually it lost, actually the enzyme activity is suppressed. Okay, so this is the signal pathway of insulin in the brain, and how it is implicated in the learning and memory. Okay, now what is the source of brain in the source of insulin in the brain? Okay, if you see here, the origin of brain insulin is controversial. However, there are certain experiments where it has found that Drosophila, okay, Drosophila have the insulin-like peptides synthesized in the brain neurons. Whereas in some in vitro experiment in rabbit neuronal cells, it has been found that the insulin can be synthesized in those rabbit neuronal cells. But in the human neuronal cells, actually the origin of brain insulin is controversial. There is no there is a lot of research to be done whether the human brain can synthesize insulin or not. So the local and neuronal synthesis of insulin is still debatable. However, if you see the insulin levels in the cerebrospinal fluid, it is of 25% of those in the blood. Generally it observes. Now this insulin level in cerebrospinal fluid, which is almost 25% to that of blood, so we should know from where it is coming. Okay, so the possible pathway for coming, or for this insulin levels in the CSF may be existed by these two pathway. That is, a fraction of plasma insulin is able to cross the blood-brain barrier via a saturable transport process, possibly through the insulin receptor on vascular endothelium. So when there is an increase in the plasma insulin after food, and after food the insulin level is increases, so probably it can cross the blood-brain barrier through the insulin receptor, which is located in the vascular endothelium of the blood-brain barrier, and that's how it can transport it into the brain. That is one hypothesis. The second one is, some regions of the brain, such as hypothalamus, it lacks effective blood-brain barrier, okay, lacks effective barrier, allowing insulin more rapidly to cross. So if you this has been supported that if a insulin injection is given and rapid insulin injection is given, then the insulin signaling is increased in the hypothalamus. So if you see here, it is supported by, it has shown that rapid activation of insulin signaling in these regions after peripheral insulin injection. So if there is an increase in the plasma concentration of the insulin, then it has been found there is increased insulin signaling in hypothalamus. So that may be because there is a lack of effective barrier in the region of hypothalamus where the insulin can cross through the barrier and pass into the brain. So this is how the source of insulin in the brain has been recognized or hypothesized. Now, there are experiments where it has been shown the insulin receptors or insulin receptor substrate and insulin like growth factors receptors which is actually expressed in different regions of the brain. So this image shows you the expression, the gene expression of insulin receptor, insulin like growth factor 1 receptor, IRS1 and IRS2 in the brain of male mice, male C57BL6 mice. It is not in human. So if I show here, which is very much linked to our Alzheimer's disease, so I will concentrate on this two particular part of the brain. Okay, this is the region of the brain, that is prefrontal cortex and hippocampus. If you see here, the cortex and hippocampus in all the image, then you can see that the percentage of, or the copies of RNA expression from 200 to 1000, and this is 1500 to 13,000. So based on the color, if it is densely colored, then more RNA is expressed. So based on this, if you see cerebellum is very dense, so that indicates there is highly expression of insulin receptor in the cerebellum. Okay, but the density is reduced, but it is not completely abolished. So prefrontal cortex and hippocampus, they have the presence of insulin receptors. Okay, at the same times, the insulin like growth factor 1 receptors, this is very dense in cerebellum, whereas frontal cortex and hippocampus, there is presence of the insulin receptor and insulin like growth factor 1 receptor. And this is also similar to the similar observation is found for the IRS1 and IRS2. So the meaning of putting this slide is that the brain is having a significant expression of the insulin receptors, and is insulin receptor substrate 1 and 2, and insulin like growth factor 1 receptor. So what does it mean? If there is a significant expression of the insulin receptor, then definitely there is the function of insulin signaling, or there is a function of insulin in the brain. Now, is there any link between diabetes mellitus and Alzheimer's disease? If you see here, Alzheimer's disease is also called as, or termed as, diabetes of brain. Okay, if you see here, it is termed as diabetes of brain. Actually, in 2008, a researcher from Brown University, Dr. Susan and Dr. Jack, they have put forward a proposal that Alzheimer's disease could be termed as type 3 diabetes, because there is a diabetes of brain, means there is a actually disruption in the glucose metabolism in the brain. So this proposal of putting Alzheimer's disease as type 3 diabetes is not widely accepted by the different bodies, world bodies. So this is just a terminology used, but it is not officially been announced that Alzheimer's disease will be called as a type 3 diabetes. This is just a proposal. Okay. A meta-analysis study in 2016 reported that people who have type 2 diabetes may be up to 60% more likely to develop Alzheimer's disease or vascular dementia. Now, why this type 2 diabetes has the link? Because there may be a hyperinsulinemia, or there may be a hypoinsulinemia. So in this case, in both the case, whether there is increased insulin level or insulin resistance, and there is less or low level of insulin, both these causes a development of Alzheimer's disease and vascular dementia, which is up to 60% more likely. And further, the link between diabetes mellitus and Alzheimer's disease, if you see, brain insulin resistance is observed in Alzheimer's disease. And patients with type 1 and type 2 diabetes are at high risk for cognitive decline with aging. And patients with the type 2 diabetes have a higher rate and more rapid progression of Alzheimer's disease. And multiple rodent models of diabetes have shown memory impairment in the experiment. So if you know here, the diabetes mellitus is mostly due to the two major reasons, isn't it? One is insulin resistance, or the other one is the hypoinsulinemia. So this link has been shown that between diabetes mellitus and Alzheimer's disease is actually extrapolate to the link between insulin and its function in the brain in Alzheimer's disease patient. So we have seen some of the experiment we have done in our lab. What we have done, we have induced type 2 diabetes in the mouse model, and then we have isolated the brain. If you see here, we have taken C57BL6 mice, and we have administered some chow pellet, and then this is a normal mice, whereas this is a diabetic mice where we have administered streptozotocin and high fat diet, which is 60% kilocalorie, and we have developed type 2 diabetes in the mice. And then we have isolated the brain, and what we have observed in the insulin signaling pathway in the brain, we have measured the IRS1, phosphorylated IRS1, AKT, AKT2, phosphorylated AKT, PI3K, and GAPDH is the endogenous control. So if you see here, there are two parts we have taken: frontal cortex and hippocampus. And based on this, the green color is actually the frontal cortex, and the white is actually the hippocampus. So if you see here, the AKT1, AKT2, and phosphorylated, especially the phosphorylated AKT, it is significantly reduced, whereas the IRS1 and phosphorylated IRS1 and PI3K, which is very important signaling protein of the insulin, has been reduced in the diabetic frontal cortex. So this NFC means normal frontal cortex, and DFC means diabetic frontal cortex. Similarly, for hippocampus, we have observed there is a significant reduction in IRS1 and phosphorylated IRS1, and also phosphorylated AKT in the hippocampus. Although we didn't find any other changes in the hippocampus, but this significant changes in this insulin signaling pathway, it suggests that there is a impairment in the insulin signaling in the brain. Now, link between insulin and brain in the Alzheimer's disease. So already we have talked about a lot about the diabetes mellitus and the brain, okay, or Alzheimer's disease. So now we will discuss about the connection between insulin and the different pathological hallmarks of the Alzheimer's disease patient. So all of you know what is Alzheimer's disease, so I don't need to explain. This is a very common neurodegenerative disease, and Alzheimer's disease is definitely you find in two types. One type is called as sporadic, another is familial. The sporadic is you will find in the patient whose age is more than 65. So as the patient get aged, you will find the sporadic Alzheimer's disease. And if the patient is less than 65 years old and getting the Alzheimer's disease, then it is linked to the genetic condition, and that will be called as familial Alzheimer's disease. So in case of sporadic Alzheimer's disease or familial Alzheimer's disease, the pathological hallmarks of sporadic Alzheimer's disease patient, if you see, it is the development of amyloid beta, development of tau proteins, cholinergic pathway, glutaminergic pathway, and then there are other pathway like oxidative stress, mitochondrial dysfunction, and neuroinflammation. These are the major pathway which are considered to be the pathological hallmarks for the development of Alzheimer's disease. So let us discuss one by one about the link between insulin and the pathological hallmarks. So first we'll discuss about the link between insulin and amyloid beta production. So this picture, if you can see here, this is called as the amyloid pathway is basically two types. One is non-amyloidogenic pathway, another is amyloidogenic pathway. So if you see here, amyloid plaque consists of an amyloid precursor protein. So this APP, actually it is a normal protein which is produced in our brain, amyloid precursor protein. But if you say 95% of amyloid precursor protein undergoes non-amyloidogenic pathway, whereas 5% of amyloidogenic APP protein actually undergoes amyloidogenic pathway. So if the A beta is formed in the normal brain, normal human, then this A beta is immediately cleared by certain cells like glial cells and microglial cells. Okay, so in case of Alzheimer's disease, what happens? In case of Alzheimer's disease, actually the amyloidogenic pathway become more evident compared to non-amyloidogenic pathway. So first, we'll discuss about the normal pathway, that is non-amylogenic pathway. What happens? The APP is cleaved by the alpha secretase at the uh, at the uh, uh, at the N terminal or C terminal, actually, in the middle of the APP, and it makes one, that is N terminal, which is called a soluble amyloid precursor protein alpha, and the C terminal is becomes C terminal fragment alpha. So alpha secretase is a normal enzyme that act on the APP and cleave it to N terminal and C terminal. And further, the C terminal fragment alpha is converted to AICD, is cleaved to AICD. This is called as amyloid APP intracellular domain, and also the P3. If you see here, this is the P3, and P3 is a very important component for the neuronal neuroprotective activity. So this is non-amylogenic pathway where APP undergoes up to the formation of AICD, that is APP intracellular domain, and P3. P3 is very important for neuroprotective purpose. But in the other way, when it is amylogenic pathway, then the beta secretase is activated instead of alpha, and beta secretase, it cut the APP uh, actually towards the N terminal, towards more towards the N terminal. If you see here, the length of the S APP alpha and S APP beta, S stands for soluble APP beta, then the S APP alpha is longer one than the beta. So beta secretase, actually it cut the APP more towards the N terminal and making the S soluble APP beta. And the other one is it become the C terminal fragment beta, which is longer than, which is whose length is more than the normal CTF alpha. And afterwards, the same enzyme alpha secretase act on that, on the CTF beta, but now it cut the CTF beta into AICD just like the normal one, but the other portion, instead of P3, it becomes A beta. Now A beta is a longer, having a longer amino acids, and this A beta actually having easily accumulated in the extracellular portion of the neurons, and then that can be actually attract the macrophages or phagocytic cells, and resulting in the inflammation of the neuron. And this A beta actually is very fibril, means very sticky together. So when there is lot of A beta is formed, then they stick together and then they form the amyloid plaque. And once the amyloid plaque is formed, then our body, the phagocytes, will try to remove them. So it attacks on the amyloid plaques and try to remove them, but that results in the formation of inflammation, means triggering the inflammation. And due to this inflammatory response, that may cause the blockade of cell to cell communication or neuronal communication, and further increase in the oxidative stress, so the neurons is further damaged. So there are two type of A beta is found, actually. If you see here, it is based on the length of the A beta. One is A beta 40 and another is A beta 42. And in between A beta 40 and A beta 42, A beta 42 is considered to be more fibrinogenic than A beta 40, and it is one of the key component of amyloid plaque. So this figure shows you how the amyloid plaque is deposited in the extracellular portion of the neuron. Okay, so this red color, the red color is considered to be the amyloid plaque, okay, in the neuron. Now, what is the link between insulin and A beta? If you see here, as I informed that A beta is actually it is cleared from the brain by different cells, especially the microglial cells and astrocytes. Okay, and during that process of clearance, this A beta actually is cleared well in a normal brain, whereas in Alzheimer's brain, it is not been degraded, or it's not been cleared completely, and that may result in the aggregation of A beta. Okay, now how insulin can influence on the clearance of the A beta? So if you see here, A beta and insulin is degraded by a common enzyme, that is insulin degrading enzyme in the brain. So when it comes to the brain, the degradation of insulin is due to the insulin degrading enzyme in the brain. And actually, this insulin degrading enzyme, IDE, if you see here, it degrades insulin as well as A beta. And this enzyme is actually released by the microglial cells. Okay. Now, in case of hyperinsulinemia, what happens? A lot of insulin occupy the IDE enzyme, making little availability for the A beta. So since lot of in case of hyperinsulinemia, lot of insulin is present in the brain, so that will occupied by the insulin degrading enzyme and making more available for insulin for the degradation. But at the same time, the amount of insulin degrading enzyme available for A beta clearance is reduced. So what you find, the IDE enzyme level for A beta clearance is reduced, and due to that, A beta is not cleared well, and that will be actually form the plaque, or they will be aggregated. And similarly, another process is central insulin resistance that causes a decrease in IDE expression also. One more mechanism is if there is insulin resistance that causes the decreased expression of IDE, insulin degrading enzyme. So both these actually causes the decreased degradation of A beta and accumulation of A beta, resulting in A beta plaque formation. Okay, this is an example. This is an experiment where it shows that how the A beta IDE actually causes A beta degradation, and in presence of insulin, what happens to the AD beta degradation. So this, if you first see the figure A here, then you will find that in presence of ID, that is, one is the normal mice where the ID is expressed, and another is the knockout mice where the ID enzyme is knocked out. Okay, this is the knockout model. So if you see here, the in presence of in a normal mice, the IDE is available, and that resulted in the A beta degradation. So you will find higher degradation of A beta. And in case of knockout mice, the degradation of A beta is significantly reduced. If you see here, it is very significantly reduced. Okay, but let us see the same thing in insulin hyperinsulinemic conditions where we put insulin. So when there is a hyperinsulinic conditions where we put insulin, administered insulin to the brain, then what we found is, although there is the IDE presented in a normal mouse, but the A beta degradation is reduced. So if you compare between normal where there is no insulin and the normal where there is insulin present, then you will find in presence of insulin, the A beta degradation is reduced, and it is significant. Although the knockout mouse, it doesn't affect it much. So what does it indicate? That in presence of insulin, what happens? Insulin binds to the ID, and that results in the lack of availability of ID for A beta degradation, and you will find less degradation of A beta. Now coming to the B's here, the figure B, you will find there are neuronal culture where IDE is being administered or treated, and it has been checked the percentage of A beta degradation. So this one, the top one, is neuronal culture with IDE, where you find the percentage of A beta degradation is reaching until 75%, whereas the second one is the A beta degradation without IDE in a neuronal culture. So the degradation is very slow, only reaching near to 25%. So if you see here the difference, it is significantly increased in presence of ID in a normal neuronal cells. Now coming to the A beta concentrations available of A beta concentration in the normal mice and in the ID knockout mice, then you can see that A beta 40 concentration in ID knockout mice after 7 days and after 14 days is significantly higher. And similarly, A beta 42 after 14 days is significantly higher. So that indicates that IDE is a very important enzyme for degradation of the A beta, and insulin actually compete with A beta for its degradation. So if you increase the insulin concentration, then that will occupy the insulin degrading enzyme, and that may result in decreased A beta degradation or clearance. So that is the conclusion from this particular slide. Now, the link between insulin and tau hyperphosphorylation, because tau protein hyperphosphorylation is a very important mechanism or mechanism of the Alzheimer's pathophysiology. So, what is tau protein? Actually, tau proteins are called as microtubule proteins, and they help in the building of microtubules of the neurons. So, tau proteins are generally very much important for building the neurons or for synthesizing or making the neurons. Okay, but what happens when the tau protein is hyperphosphorylated? So if the tau protein is hyperphosphorylated, then that can cause the degradation of the neuronal microtubules or breaking down of the neuronal microtubules, and then actually it causes the neuronal damage. So if you see here in the picture, there are two pictures. One is the normal, where you find the normal neurons and there is the size of the brain, but in the Alzheimer's brain section, you find there are degraded neurons where the neuron is degraded, and there is a shrinkage of the brain also in Alzheimer's brain. So this actually resulted because the neuron is being degraded very rapidly, and actually the architecture of the brain is actually changed here. Now let us discuss what is the function of tau protein, okay, and how it is influenced in the Alzheimer's disease. Okay, so this picture shows you the normal function of tau protein. This is the microtubules where the tau is attached with the microtubules, and it helps in the building up the microtubules. So for that process, actually the tau protein has to phosphorylate and dephosphorylate to build the microtubules. So when the tau is in the dephosphorylation stage, then the tau protein bind with the microtubules and helps in the building. And when it is phosphorylated, then the tau protein is disengaged from the microtubules and allowing the cargo for building the microtubules. So this process actually undergoes in a normal brain, the phosphorylation and dephosphorylation of the tau protein, and that helps in the building of microtubules. Now, in case of Alzheimer's disease, what happens? In case of Alzheimer's disease, this this microtubules, tau protein, actually hyperphosphorylated. So this picture, if you see here, healthy neuron, how the tau protein is actually help in the building the microtubules. But when it is hyperphosphorylated, normal phosphorylation is required, but when it is hyperphosphorylated, then it doesn't engage with the microtubules, and they will form a aggregation. Okay, if you in this case, the tau proteins is actually completely disengaged from the microtubules, and they do not bind with they could not undergo dephosphorylation, and those microtubules are aggregated, and because of aggregation of the tau proteins, the microtubules has been degraded, and later this aggregation of tau proteins leads to the formation of neurofibrillary tangles. This is called as neurofibrillary tangles. So tau proteins forms the neurofibrillary tangles in the Alzheimer's brain, and that causes the damage of neurons in the Alzheimer's brain patient. What is the link between insulin and tau hyperphosphorylation? Actually, if you see here, the insulin like growth factor 1 and uh, insulin receptor substrate 2 knockout mice have increased tau hyperphosphorylation, that has been reported, and reduced IRS1 and IRS2 also lead to hyperphosphorylation of tau. So these are all linked to the insulin signaling. So if there is a reduced insulin signaling, it has been found that hyperphosphorylation of tau protein takes place. Now, why this hyperphosphorylation of tau protein has takes place? Because it is mostly regulated by the GSK3 beta. So if you remember in my, I think, couple of slides before, I have told you that insulin actually deactivate the GSK3 beta enzyme action by phosphorylating the GSK3 beta. So if there is a lack of insulin sensitivity, then deactivation of GSK3 beta do not takes place, and the enzymatic activity of GSK3 beta stimulate the tau hyperphosphorylation. That's how the relation is. So we'll see the link in the next slide. So that's this is the link between the insulin and the into the tau hyperphosphorylation. So this is a normal process when the normal brain actually, where insulin binds and it undergoes tyrosine phosphorylation, stimulate the PIP3, then AKT, and AKT actually inhibit the conversion of inactive GSK3 beta to activated GSK3 beta. This is the process. So since the activated GSK3 beta glycogen synthesis kinase 3 beta is not there, so it doesn't actually phosphorylate the tau protein or hyperphosphorylate the tau protein. Okay, this is the normal one. Now when there is resistance, okay, when there is a insulin resistance, what happens? The insulin resistance actually will block all the insulin signaling, and it will, there is no activation of AKT, phosphorylation of AKT, and that will actually cause conversion of inactivated GSK3 beta to activated GSK3 beta. And once the GSK3 beta kinase, the kinase is activated, and due to this enzyme activity, the tau is phosphorylated, and then neurofibrillary tangles has developed, and that leads to the formation of AD. Okay, and further, the in the insulin resistance brain, there is also phosphorylation of serine moiety of the IRS1. So in a normal brain or normal insulin signaling, tyrosine phosphorylation is very much important. But if IRS is phosphorylated at the serine residue, then actually it causes a blockade of normal insulin signaling, and it start another signaling pathway. So phosphorylation of serine of the IRS1 moiety that stimulate the activated GSK3 beta, and further it causes the hyperphosphorylation of tau. So this is how takes place in a Alzheimer's in a brain who has the insulin resistance. Okay, in a insulin resistance brain actually. Now next we will find the link between insulin and cholinergic function. So all of you know what is the role of cholinergic function. It is a very old hypothesis, cholinergic hypothesis, where it has been found that there is a lot of cholinergic neuron degeneration in the basal forebrain in AD patient, and loss of cholinergic neurotransmission in the cerebral cortex and other brain regions are found in AD patient. And now effectively, if you see here, uh, uh, acetylcholinesterase inhibitors, okay, there are three acetylcholinesterase inhibitors. They are donepezil, galantamine, and rivastigmine. They are approved by US FDA for the treatment of Alzheimer's disease. And at the same time, you will find there is a deficit of choline acetyltransferase in the neuron of Alzheimer's disease patients. So if it's based on the cholinergic hypothesis, you find there is loss of cholinergic neurons. We found there is loss of choline acetyltransferase, which is responsible for synthesis of acetylcholine. This enzyme is actually reduced, and also when we use the acetylcholinesterase inhibitor, because the acetylcholinesterase is the enzyme which degrades the acetylcholine. When we use these inhibitors, then we can treat the symptoms of Alzheimer's disease. So this is the hypothesis of cholinergic hypothesis of AD patient. Now, what is the link between insulin and cholinergic function? Actually, in some experiment, it has been found that insulin increases the cholinergic transmission by augmenting the acetylcholine synthesis. So for acetylcholine synthesis, you need two important steps. First step is the choline must be transported into the neuron, and second step is the choline must be utilized by the choline acetyltransferase for the synthesis of acetylcholine. So what it has been found in the experiment that both these steps, the choline transport into the brain is also increased by insulin. So the choline uptake into the brain is increased, which is a rate limiting step of the acetylcholine synthesis. At the same time, it has also been found that in the hippocampus region of mice, insulin signaling related proteins are co-located with choline acetyltransferase. So there is no direct relation has been found, but the insulin signaling related proteins which is co-located with choline acetyltransferase enzyme may give a relation between these two, where insulin signaling may augment or improve the choline acetyltransferase enzyme function. Okay, so this two actually has been studied. Okay, and the other one is the coexistence indicate that insulin signaling may play an important part in activities of cholinergic neurons as well as. So based on this, there are some results has been published by some researcher. So what I have shown you here is the presence, first of all, if you see here, inhibition of PI3K kinase by PI3K kinase inhibitor, this LY 294002 alters choline uptake in the neuronal cells, SHSY5Y neuronal cells. So there are four figures here. So first we'll discuss the figure A. So the figure A describes choline uptake by choline acetyl transporter, which is measured as following the 5-minute exposure of differentiated neuronal cells in presence of the PI3K inhibitor. Okay. All of you know what is the role of PI3K. I told you in the insulin signaling pathway, PI3K is very important because it phosphorylates the phosphorylation of PI3K leads to phosphorylation of AKT. Understand? So the downstream signaling process. Now, if you block the PI3K, then definitely you are blocking the insulin signaling. So in the first figure A, if you see here, in presence of 0 means there is no PI3K inhibitors, and 10 and 50 means the amount, the concentration of the PI3K kinase inhibitors present in the neuronal cells. So when we see there is in absence of PI3K kinase, the choline uptake is 100%. Okay, in the control, that is 100% uptake of choline takes place in the neuronal cells. But when you add the PI3K kinase inhibitor, PI3K inhibitors, then it is slowly and dose dependently reduced. So what does it indicate? That PI3K inhibitors inhibit the choline uptake. So the insulin signaling, if you disrupt the insulin signaling, then there is inhibition of choline uptake. So insulin resistance definitely is correlated with this particular observation. Similarly, in the B, levels of uh, PKB, phosphorylated AKT or protein kinase B in the brain. So in absence of the PI3K inhibitors, the phosphorylated AKT is expressing because this is a normal, it is 1. Okay, and in presence of the PI3K inhibitor, actually the protein kinase expression is reduced. The phosphorylated protein kinase expression is reduced. So the phosphorylation of protein kinase A or AKT is a very, very important step in the insulin signaling. Okay, so if you disrupt the insulin signaling, then definitely you find there is disruption in the AKT pathway. So these two observation actually suggest that if there is normal insulin, that just like in the control, then you have 100% choline uptake and you have higher amount of phosphorylation of AKT or it is protein kinase B. But if you disrupt the insulin signaling, then the choline uptake is reduced, and similarly the phosphorylation of the downstream signaling protein is reduced. So this is what has been observed by the different researchers. Now next we will have the discussion between insulin and glutamate excitotoxicity. So glutamate is considered to be one of the another important pathway where it causes damage of the neurons, because glutamate is considered to be one of the excitatory neurotransmitter. And when the excitatory neurotransmitter glutamate binds to its receptor, that is NMDA receptors, that causes an hyperexcitability and induce calcium overload. Anyhow, for normal function, actually a certain amount of excitation is required. But if it is hyperexcited due to excessive glutamatergic function, then that may cause an increased overload of calcium, And calcium overload, actually, you know that it can lead to depolarization of the cells, and complete depolarization or prolonged depolarization of a cell will lead to the actually the damaging of the cell, or it is even the death of the cells. Okay, so if there is a excessive glutamatergic function or hyperexcitability of NMDA receptor, that can induce calcium overload, that can cause excitotoxicity, mitochondrial depolarization, and since mitochondrial depolarization can lead to mitochondrial dysfunction, so you will find there is a ATP depletion and also the neuronal death. And it has been also found that NMDA receptors are directly or indirectly regulated by A beta. So if there is a accumulation of A beta, then you may find the glutamate function is increased, and you may find there is a hyperexcitability of NMDA receptors. And the drug memantine is another drug which is used for the treatment of Alzheimer's disease, and it is a NMDA receptor antagonist and approved by the US FDA. So this is the normal pathway of glutamate excitotoxicity in the development of Alzheimer's disease pathophysiology. Now, what is the link between insulin and glutamate excitotoxicity? There are controversial reports on insulin and glutamate response, and researchers suggested that insulin potentiates the effect of glutamate, leading to cellular death. But at the same time, another group of researchers reported that insulin attenuates the glutamate hyperexcitability and protect the neurons. So probably this controversial, if you see here, the observations are just opposite to each other. So this opposite the result or controversial results may be due to the concentration of insulin used and the duration of insulin is used. So based on that, probably the effect is less. Probably if you use insulin with a very low concentration, if you use insulin as a very low concentrations and for very acute period of time, that may cause that may cause attenuation of glutamate hyperexcitability. But if you use the insulin for a prolonged period of time with high concentration and develop like a hyperinsulinemic conditions, then probably that may potentiate the glutamatergic excitability and cause cellular death. So that is been not identified yet. So you just a hypothesis, understand? And recently, a group of researchers reported that short-term exposure of insulin protected the cultured cortical neurons against excitotoxicity by attenuating the glutamate-evoked rise of intracellular calcium and preventing the onset of delayed calcium dysregulation. And also, it improved the oxygen consumption rate and oxidative mitochondrial metabolism, and increase the brain derived neurotropic factor. So its short term exposure probably has led to the protection of the neurons in this research has been described. So let us see the result of those. If you see here, the cell viability of the neuronal cells, these are all in vitro experiments, so these are not in vivo. So in a cellular, if you see here, the cell viability of the neuronal cells, the control is 100%. Definitely it is 100% because there is no treatment. And in presence of insulin, this is also 100% cell viability. So cells are is more active, means there is cell death is zero, considered to be zero. And when you use the glutamate in a certain concentration, the cell viability is reduced below 50%. It is almost coming towards 30 or 40%. So it is a significant reduction in the cell viability. So that indicates that cells are damaged, or the cells are died because of glutamate. But when you add insulin to that group where you added the glutamate, so it is glutamate plus insulin, you will find the cell viability is significantly increased. So this indicates that the insulin protects the glutaminergic toxicity, and that's why the insulin cause an increased cell viability. So this is the report has been published recently. And now another report which is published recently, I think it's 2019, is if you compare with the BDNF level, okay, brain derived neurotropic factor, which is a very important neuroprotective protein. found in the brain, this is also correlate with the cell viability result. So in case of control in insulin, the the concentration is very high, but when you add the glutamate, the concentration of BDNF is reduced, and in presence of insulin, the concentration is significantly increased. Now, why the reasons of this cell viability and BDNF? If you see the picture here, the fluorimetric responses, one, this response is the intracellular calcium concentration. If you see the intracellular calcium concentration of the glutamate, then you find it is almost coming to 8 AU or more than 8 AU. But in presence of insulin, the glutamate can only induce up to 6 AU. And the picture, the figure, it shows a very clear, it demonstrates a very clear difference between the glutamate, which is the red in color, and the glutamate with insulin, which is the blue in color. Okay, so there is a significant reduction in the intracellular calcium in the neuronal cells in presence of insulin. So the calcium overburden is reduced. At the same time, the membrane potential of the neuron, if you see, in presence of glutamate, the membrane potential is almost reached to 6 AU or more than 6 AU. But in presence of insulin and glutamate, the membrane potential of the mitochondria is reduced to below 6. And if you see the compiled picture here, the red color one is very high, which is only glutamate, whereas the blue color is glutamate with insulin. So that indicates that the presence of insulin that leads to decreased intracellular calcium overload. At the same time, it decreases the mitochondrial membrane potential. So both these actually protect the neuronal cells from its damage. One another, they have also expressed the mitochondrial functions in terms of measuring the oxygen consumption rate and percentage of respiration and ATP. So if you see here, the oxygen consumption rate is significantly improved in the insulin treated group, that is the blue in color, where it is decreased in the glutamate. And the mitochondrial respiration is also increased in presence of insulin, whereas the ATP formation is also increased dose dependently in presence of insulin. So what does it indicate? That actually insulin protects the mitochondrial function, which is showed here by improving in the oxygen consumption rate, improving in the respiration, and improving in the ATP formation. Now let's have the link between the oxidative stress, neuroinflammation, and mitochondrial dysfunction, the insulin role. So actually, as we have already discussed, the brain is the highly metabolically active organ. So it is very vulnerable to the oxidative stress, and there is a strong link between oxidative stress, neuroinflammation, and mitochondrial dysfunction has been provided. So this is generally, we cannot say actually neuroinflammation can lead to oxidative stress or mitochondrial dysfunction. Actually, they are all interrelated. They are all interrelated, just like egg and chicken. Okay, so that may lead to neurodegeneration. So these are the normal healthy brain where there is a high oxygen consumption by the neurons, high content of pro-oxidant metals you will find, high polyunsaturated fatty acid content, and low antioxidant defense you will find in a normal brain. And whereas in a neuroinflammatory insulin resistance brain, you will find all these is disturbed. So you will find increased oxidative stress, apoptosis, and protein aggregations, mitochondrial dysfunction, all will be observed in a insulin resistance brain. So in an insulin resistance brain, actually it can trigger all this due to the mitochondrial dysfunction or due to the burst of reactive oxygen species or oxidative stress that stimulate the inflammasomes, and you get a lot of inflammations or formation of all these inflammatory mediators. Okay, and these inflammatory mediators also have an effect on the insulin signaling. So if you see in our next slide, I'll explain you how the inflammatory cytokines cause insulin signaling disruption of the insulin signaling. So overproduction of ROS and ATP depletion, activation of pro-inflammatory cytokines, all these cause the neuronal damage. And Okay, increased expression of cytokines. Yeah, it causes all inflammatory damage. Okay, so how the inflammatory cytokines can cause disruption of the insulin signaling? If you see here, so this is the TNF alpha, one of the inflammatory cytokines. It binds to its receptor and causes the stimulation of JNK pathway. And JNK pathway actually it causes serine phosphorylation instead of tyrosine phosphorylation. So the serine phosphorylation actually negatively regulates the insulin signaling, whereas tyrosine phosphorylation it positively regulates the insulin signaling. So serine phosphorylation definitely it causes a disruption of the insulin signaling, and because of that you will find impaired glucose transport and also loss of normal neuronal hemostasis and synaptic plasticity role. So this how the inflammatory markers or cytokines can influence the insulin signaling in the brain, and that can cause the impairment of the glucose metabolism and also impairment of the normal neuronal functions. So we have found some of the inflammatory markers in the diabetic mice, the brain of the diabetic mice, and we have found there is a significant increase in the inflammatory markers IL-6 and IL-1 beta in the hippocampus, diabetic hippocampus. Okay, and even in the frontal cortex, the blue one. Whereas we have found significant decrease in the SOD1, superoxide dismutase, which is a very important enzyme in the mitochondria for scavenging the free radicals. So SOD1 is significantly reduced in the diabetic frontal cortex and diabetic hippocampus. So this is what we have observed now, how it has been influencing the mitochondrial function in a insulin resistance brain. So in insulin resistance brain, actually decreases the brain mitochondrial electron transport chain and also reduce the mitochondrial respiration and also reduce the mitochondrial DNA synthesis, so which is linked to the mitochondrial biogenesis. And it has been also observed that there is a disturbance in mitochondrial fusion and fission in both in vitro and in vivo animal models. So insulin resistance has a direct link on the mitochondrial function, mitochondrial degeneration, and mitochondrial biogenesis. So how it is linked, actually, if you see here, actually the all this is linked through the insulin or the energy deprivations. So this is all through the AMPK pathway, where it stimulates the PGC1 alpha, and C-twin is also stimulate the PGC1 alpha, and PGC1 alpha actually it nuclear that is the respiratory factor is stimulated, and that causes mitochondrial DNA and mitochondrial biogenesis, improved mitochondrial biogenesis. And similarly, PGC1 alpha also stimulate the mitochondrial respiration, and that's how the mitochondrial function is increased. And somehow the this one AMPK also regulate the GLUT4 translocations, and even the AKT regulate the PGC1 alpha. Okay, so that's why the insulin signaling can regulate the PGC1 alpha, and then further it regulate the mitochondrial function. But in case of insulin resistance, what will happen? There is no signaling, so you will not find the link between AKT and PGC1 alpha, so your mitochondrial function is reduced. Okay, so we have found the difference between the PGC1 alpha, because this is a central regulator of the mitochondrial function. PGC1 alpha is a very important marker for understanding the mitochondrial biogenesis and mitochondrial function. And we have also observed the AMPK phosphorylate AMPK, total AMPK, and phosphorylated AMPK. What we found is in the diabetic hippocampus and diabetic frontal cortex, the PGC1 alpha is significantly reduced, indicating that mitochondrial function is going to be reduced, or it is reduced in the brain. Whereas we didn't find any link on AMPK. There is no much change in the AMPK pathway. AMPK. So this PGC1 alpha probably is regulated by the insulin, as I told, the insulin pathway. That's why, since there is a insulin resistance, so you will find there is decrease in the PGC1 alpha, but there is no change in the AMPK pathway. So we don't have any change in the AMPK pathway. That's why the observation is. Now the key point, let us summarize the lecture today, the talk today. No report confirmed insulin synthesis in human neuronal cells or brain. Most brain regions have insulin resistance and the change in the insulin growth factor receptors, okay, in a normal brain. But in case of insulin, it facilitates the A beta degradation through the IDE, insulin degrading enzyme. It attenuates the tau hyperphosphorylation through the GSK3 beta inactivation. It stimulates the choline uptake. It protects the glutamate induced excitotoxicity. And in insulin resistance brain, we found there is increased pro-oxidant enzymes and reduced antioxidant enzymes and increased inflammatory markers and increased serine phosphorylation of the insulin signaling IRS protein. Okay, so mitochondrial dysfunction is also been observed. So if you see the key point, the last point, what you can take home message, the insulin deficiency, hyperinsulinemia, and insulin resistance and dysregulated insulin signaling pathway, all is linked in the brain, is all linked to the development of Alzheimer's disease pathophysiology. So this is what the take home message for today's talk. These are my references. And this is my team. We have a lot of they have helped me in this research. So thank you for your attention, and have a good day. Any questions, please? So I end my talk here. Yes. Hello. Biswa, Dr. Biswa.
Yes, yes, sir. Some questions on chat box, sir. We will discuss.
Sorry.
Sir, we
Can
have
you?
received some questions on chat box.
Oh, chat box. Where it is?
I'll show, sir. I'll show.
Okay, okay. I got some question. Yeah, I got the question. Yeah. Yeah. Let me see. Let me see the questions. Okay, the one question is, can administration of insulin can protect the person for producing Alzheimer's disease? Okay, this is actually, if you see, there are now available insulin intranasal formulations for the treatment of Alzheimer's disease. So definitely, if you administer insulin, it may protect the Alzheimer's disease as well. It is already in the market, I think. Yeah. How much units of insulin used that can be protect the neuron from glutamate excitotoxicity? This is a concentration dependent questions. As I told, that's one of the researcher has shown in short term exposure of insulin that causes a protection of glutamate excitotoxicity by inhibiting the increased intracellular calcium or by inhibiting the calcium overload. So that I think you can refer the article which is present, which is there in the reference, the concentration, how much concentration they have used. And the other questions are, please inform me the dose of insulin that at which protects the neuron from glutamate. I think a dose of insulin need to be validated in your lab, and also you refer those articles which is published. They have used different concentration of insulin either in vitro or in vivo. So based on your experiment, you can choose your dose, whether it will be in vitro or it will be the in vivo experiments. And what are yes, there is a very important that because insulin produces hypoglycemia. Yeah, that's true. So you need to be carefully choose the insulin for the treatment of for this particular experiment if you are using in vivo experiment. Otherwise you can this can be better observed in an in vitro experiment where you have no problem with the hypoglycemia. Okay. What are the causes of insulin resistance? How to overcome the insulin resistance? Insulin resistance causes is, all of we know that lipid, hyperlipid means there is obesity is one of the cause where the lipid is deposited in the muscle cell, and due to excessive lipid deposition, that is called as lipotoxicity, your expression of GLUT4 to the skeletal muscle cell is reduced. So once the GLUT4 translocation is reduced, then your glucose utilization is reduced. And this is also linked to genetic condition also, where you have the expression of insulin receptor is reduced. So if the expression of insulin receptor is reduced, then that will also cause insulin resistance. So in both the way, the insulin resistance cause. One way where the insulin receptor is expression is reduced, and that may be linked to your genetic condition. The other way, it is due to the obesity, where the lipid is deposited in the skeletal muscle and cause lipotoxicity, and because of lipotoxicity, GLUT4 translocation do not take place, so glucose is not utilized. Okay, that's the main that's the answer for this. Okay, so any question?
Thank you, sir, for your nice presentation. So you have
Okay.
really correlated this insulin and also the effect in relation to the Alzheimer's disease. So it will be beneficial to our research scholars and also faculty members who are attending this program. So we are very much thankful to you, sir, for sparing your valuable time with us, sir.
Okay, thank you so much. So good afternoon, Bimal. Yes, good afternoon, Bimalendu. Yeah, fine, fine. Oh, you are with Bispamohan because the name is showing Bispamohan only.
Yes, yes.
Bimalendu. Yeah. Okay, okay. Okay, good. How are you, Bimalendu? Can you hear me? Hello.
Sir, unmute किया नहीं.
Okay. Bimalendu sir, tell me.
Yes, sir. Yes, sir. Where you are coming from, Orissa?
Orissa, there is no confirmed date.
Again, the corona pandemic is increasing day by day.
Yes, Corona pandemic. This time more than
sir.
1 year. Yeah.
Sir, any new vaccine from Malaysia, sir?
Sorry?
Any new vaccine from Malaysia? We send.
New vaccine? No new vaccine. They are all taking the Sinovac, China one, and Pfizer. Pfizer's what is that? Pfizer vaccine. That's all.
Covishield.
They are taking these two only. Okay.
Thank you so much, sir, for this insightful session. I now request our respected principal, sir, Dr. V.V. Ravi Kumar, sir, also the coordinator of this FDP program, to kindly present e-certificate of appreciation to Dr. Subrat Kumar Bhattamishra, sir, as a part of token of respect and gratitude from Roland Institute of Pharmaceutical Sciences, Barampur.
Thank you, Dr. Subrat, for accepting our invitation and sparing valuable time with all of us. I'm very sure that your talk will be definitely useful for all the participants who participated today. And I want to acknowledge your contribution to Roland Institute of Pharmaceutical Sciences. Because of people like you, Roland is still in the #1 position in pharmacy field education. So once again, I thank you for sparing your time. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Kindly accept it, sir.
Okay, thank you so much, sir. Thank you so much. Yes.
Okay, thank you, sir. Bye.
Okay. Bye bye.
Meet you at Barampur.
Have a good day. Okay. 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 See you. Okay. Bye, sir. Bye.